Hello students, today we are going to discuss about one of the practical, that is water quality analysis by a method known as MPN, or most probable number. Like we have discussed earlier, we have other methods as well, like membrane filtration method and some other methods, but today we are going to discuss about the most probable number method. So this method, or the most probable number analysis, is a statistical method based on the random dispersion of microorganism per volume in a given sample. So in this method, the measured volumes of water are added to a series of tubes, as you can see the picture right on the diagram. So a measured volume of water are added to a series of tubes containing a liquid indicator growth medium. Then the media receiving one or more indicator bacteria will show growth and a characteristic color change. You will observe the color change. Then this color change is absent in those which receive, or in those receiving only an inoculums of water without indicator bacteria. Then from the number and distribution of positive and negative reactions, the MPN of indicator organisms in the sample may be estimated by reference to the statistical tables. So coming to the different again another steps for this MPN. So this MPN test is completed in 3 steps. So the first is presumptive test, then we have complete test, and the last one is the second is confirmed test, and the third last one is the complete test. So we are going to see about each and every of these steps. So the first is the presumptive test, right? So out here, this test is a specific enrichment procedure for coliform bacteria, which is conducted in fermentation tubes filled with a selective growth medium, like example McConkey lactose broth, which contain inverted Durham's tube inside for the detection of fermentation of gas, right? Then a series of lactose broth tubes are then inoculated with measured amounts of the water sample to be tested. So the series of tubes may consist of 3 or 4 groups of 3, 5 or more tubes. So the main selective factors found in the medium are lactose, sometimes a surfactant such as sulfate or bile salt, and often a pH indicator dye for facilitating detection of acid production, such as bromocresol purple or brilliant green. And the selective action of lactose will occur because many bacteria cannot ferment this sugar, whereas coliform bacteria and several other bacterial types can ferment this sugar. So the surfactant and dye do not inhibit coliform bacteria, whereas many other bacteria such as the spore formers are inhibited. So this is for the presumptive test. Okay, so here you will see we have the conical flask that contains the original sample of pond water. Out here we used a sample of pond water. Then we will transfer 10 ml to 5 test tubes, and another 1 ml to another 5 test tubes, and 0.1 ml to another 5 test tubes. So we will inoculate all this pond water in this different sets of test tubes, 3 sets. In one set we have 5 test tubes, right? Then after that we will incubate all these test tubes at 37 degrees Celsius for around 24 hours. Then after that we will check for the whatever we want to see, like whether gas production is there, acid produced or not. So all these things you will see after the incubation period. So in the first batch of the 5 test tubes, you will see all the tubes are positive, right? There is acid production, gas produced, and then in the second batch of test tubes, another 5 test tubes, we will see only the first 2 test tubes were positive, while the rest 3 are negative. Then in the third batch, or in the third another 5 test tubes, all the test tubes are tested negative, where there is no positive, not a single positive is present. So this is for the presumptive test. Next we have is the confirmed test. So this confirmed test, it can be done in 2 ways or 2 parts. So this test serves to confirm the presence of coliform bacteria when either a positive or doubtful presumptive test is obtained. So here a loop full of growth from such presumptive tube, it will be taken from the presumptive tube, is transferred into a tube of brilliant green brilliant green lactose bile, or we also call it BGLB, 2% broth or other lactose broth we can use. And then after that we will incubate at 35 degrees Celsius for 48 hours. Then this medium, it is a selective medium for detecting coliform bacteria in water as well as other samples like dairy products and other food products. So a selective agent in this medium is the lactose. So the broth tube also contains a Durham's tube. For the purpose is to detect the gas production. Okay, then a plate of LES endo agar, or we also call it EMB agar, is streaked with a loop full of growth from a positive tube and incubated at 35 degrees Celsius for around 18 to 24 hours. After that, typical coliform bacteria like Escherichia coli and Enterobacter aerogenes will exhibit good growth on this medium, and you will see the changes of color that will form red to black colonies with which dark centers or sheen is present. Then the Salmonella typhi will exhibit good growth, but the colonies are colorless. Like another example of organism we have is the Staphylococcus aureus, where the growth is inhibited altogether. So that's all for the confirmed test. Next we have is the completed test. So this test, it helps to further confirm doubtful and, if desired, positive confirmed test results. So a typical coliform colony from an LES endo-agar plate is then inoculated into a tube of brilliant green bile broth and on the surface of a nutrient agar slam. Then they are incubated at 35 degrees Celsius for 24 hours. Then after 24 hours, the broth is checked for the production of gas and a gram stain is made from organisms on the nutrient agar slam. So if the organism is a gram negative non-spore forming broth and produce gas in the lactose tube, then it is positive that coliforms are present in the water sample. So that is for the completed test. So what are the uses of this MPN, or we also call it most probable number? So this uses includes, it is commonly used in estimating microbial population in soil, waters, agricultural products and some other samples as well. Then we have this another uses that includes, this technique is useful in particular samples that contain particulate material that interferes with plate count enumeration methods, and it is also useful because it is suggested as a consideration for an alternate method to strengthen environmental monitoring studies. So that is one of the uses. Then also it is useful for counting bacteria that reluctant form colonies on agar plates or membrane filter but grow readily in liquid media. So that these are the few uses we have here for the MPN. Then the advantages of most probable number. So what are the advantages we have? The advantages is that this MPN, it is ease of interpretation either by observation or gas emission. Then the sample toxins are dilated. Another advantage is that sample toxins are dilated. Then effective method of analyzing highly turbid samples such as sediment, sludge, mud, etc. Then another advantage is that it permits samples that cannot be analyzed by membrane filtration. So these are the advantages for MPN. Then the limitations, we have also few limitations mentioned. It is the limitation of this MPN, it shows poor accuracy and precision associated with MPN counts usually, which means that the method is one of last resort to be considered only when other counting methods are inappropriate, and laborious and expensive in terms of materials, glassware and incubator space, and it has a relatively large margin of error. So these are the advantages and limitations of most probable number. So before we end this topic, we will see, like I forgot to mention about the the results or the interpretation, how we will interpret or we will get the results for this different steps. So first we'll discuss with the presumptive test. Okay, so in the presumptive test, what will happen, what will be the result? In case if it is positive, the formation of 10% gas or more in the Durham's tube within 24 to 48 hours, together with turbidity in the growth medium and the color change in the medium, constitutes a positive presumptive test for the coliform bacteria and hence the possibility of fecal pollution. The test is presumptive only because under these conditions, several other types of bacteria can produce similar results. So in case if it is negative, you will see no growth or formation of gas in the Durham's tube, right? Then for the confirmed test, we have the result, it is interpreted in case of positive, you will see formation of gas in lactose broth and demonstration of a coliform like colony in the EMB agar indicate the presence of a member of the coliform group in the sample examined. Then the coliforms produce colonies with greenish metallic sheen, which differentiates it from non-coliform colonies. Okay, then you will see presence of typical colonies on high temperature, that is 44.5 plus minus 0.2. This will indicate presence of thermotolerant Escherichia coli. So that is for the case of positive results. Then in case if it is negative, you will see the absence of gas formation in lactose broth or the failure to demonstrate coliform like colonies on the EMB agar. So this is for the confirmed test. So in case of next step, the complete test, right? So in case of complete test, here in case if it shows positive, the result is positive, how will you know? So you will know by the presence of gas in the brilliant green bile broth tube and gram negative non-sporing forming rods on nutrient agar sand constitutes a positive completed test for the presence of coliform bacteria, which in turn infers possible contamination of the water sample with fecal matter. So if your results is negative, you will see absence of growth and gas formation in the broth and absence of gram-negative non-sporing rods on gram staining when we perform the gram staining. Okay, so that's all about the MPN method, or most probable number method. Thank you.